So this is a question I was hoping that in last 18 months at some point somebody would ask. So you haven't asked a direct question, but in this question I can answer with this mechanism. Imagine here we have a B cell. This B cell is a memory B cell, or it may even be a not memory B cell. It is aware of how to work with the SARS-CoV-2 spike protein, meaning it has a B cell receptor, BCR. B cell receptors are actually immunoglobulin D. So for the healthcare students here, when I meet you, remember this, I'll ask you this question. What is a B cell receptor? B cell receptor is actually immunoglobulin D. IgD acts as a B cell receptor. So here is a B cell receptor. B cell receptor, let's say this B cell knows how to connect with SARS-CoV-2 and how to make antibodies against SARS-CoV-2. Here is, let's make one more B cell here. This is another B cell. This B cell, let's say, is a little different, and it knows how to connect with, let's say, flu virus, and how to be, how to take care of the flu virus. And then there is another B cell. I just want to make some B cells. This B cell is, let's say, able to connect with something else. Let's say common cold, rhinovirus. Right? Now the question is it possible for us to activate these B cells without getting infection or without getting the vaccine? Here is a problem. Let's say we put some cytokines here. The cytokines are the ones that helps activate the cells, that are one part of the activation. So let's say we say interleukin 4 and 5. Interleukin 4, interleukin 5. These interleukins can cause B cells to become increased in number and differentiate. They can cause proliferation and differentiation. So you say, you know what? Forget about the vaccines and forget about the infection. Let's just give interleukin 4 and 5 to the person, and that would go and work on the B cells, and that would increase their differentiation and proliferation, and voila, we have a solution. Here is the delicate point that we have not discussed throughout 18 months. Only those B cells that have actively connected with the pathogen will become active when cytokine is given or spilled on them or poured on them. So look at me for a second. Imagine I'm a B cell. I'm standing here. My receptors are able to work with the SARS-CoV-2, but there is no SARS-CoV-2 attached to me. I am not bound to any SARS-CoV-2. I'm just standing here. If I'm standing here, you can pour as much cytokine on me as you like, and I will not become active. But if you have a SARS-CoV-2 standing next to me and I hold it, then I'm ready. If you now pour the cytokine on me and this receptor is engaged, then I know that you're giving me an order to attack this thing. Then I'll become active. This is called co-stimulation. This is a protective mechanism in our body. Otherwise, anytime an infection occurs, anytime cytokines are produced, every cell in the immune system will become active. We will be destroyed by our immune system. So our immune system cells only become activated when they have actively engaged with the enemy. Then you give them an order to say attack, and they'll attack. But if the soldier is just standing there and you say attack, it's not going to do anything. This is a very important concept. So now if I go back to your question, Genesis Lights, if we say, are there any other ways to boost your antibody production without getting booster to get or reinfection? So what we are saying is this, we are saying, or you are saying, can I have this B cell of mine, which knows how to work with the SARS-CoV-2, to just become active and make more B cells of its kind and make antibodies from it? And the answer is no, until it is attached with an antigen. What does a vaccine really do? There are so many myths and rumors, and basically vaccine is just offering that antigen. It's saying, here is an antigen that looks like the virus, and then in the future when the actual virus comes in, they would attack it.